The next part of 12.2 is going to talk about in more detail about mitosis and the phases. They call them the phases of mitosis. So be able to answer objective 6 here. In number 10, it allows you to either draw or describe a bunch of these things. This is vocabulary. You got to know what these things are and understand how they participate in the mitotic process. All these words, kinetochore, non-kinetochore, make sure that you find those in the book. And then finally in number 12 it says you've got to be able to compare cytokinesis, or the division of the cytoplasm, in both animal cells versus plant cells. These are both eukaryotic cells. But first I want you to concentrate on mitosis. Many of you have learned this before, IPMAT, PMAT. And the little PM in there, I'll explain that in a minute. That's how I memorized the phases of mitosis: interphase first, and then prophase, metaphase, anaphase, telophase. The PM refers to prometaphase, which we'll talk about. You got to know these things cold. You got to know what happens in each one. This is basically middle school science with a little bit of higher level detail thrown in. So you have to know these things. The big player here is going to be this item called the mitotic spindle. You can see it here in the second graphic. It's a bunch of proteins called tubulin and actin, and what these are they're like ropes, if you will, and they're going to grab onto the chromosomes and they're going to move them all around during mitosis. They're very important. You need to understand how they're made and what exactly they do. So make sure you look for that in the book. That really happens in prophase. In interphase, this G2 phase, lots of stuff is being made. These centrosomes in particular are being made, along with the chromosomes are being duplicated, but it's not till the nuclear envelope starts to break down in prophase that scientists can actually see the chromosomes and these mitotic spindles that have been created by the centrosomes in order to grab onto the chromosomes and move them all around. So here's a centrosome. Sorry, here in yellow they're talking about it. It's also called the microtubule organizing center. It's a centrosome with centriole pairs. This is going to get confusing. It always is. Too many CE words. The centrosomes have centrioles, which then migrate to either side of the cell, and they organize all these ropes that we'll call mitotic spindles. When the centrosome replicates, it forms two centrosomes that then migrate to either side, and you can see the microtubules here that are growing out of them, all these lines, and they're going to very specifically bind to individual chromosomes. During prometaphase, this is kind of the new phase that science has added to the phases of mitosis. Some of the spindle microtubules attach to the kinetochores of the chromosomes. And what kinetochores are is they are right here, if you will. There are a bunch of proteins that exist on the centromeres. So we have centrioles, centrosomes, excuse me, and centromeres. Centromeres are just the little spot, if you remember, where the sister chromatids are bound together, and it's where the chromatids are bound together as well as where the mitotic spindles attach. You can see that right here. It's very important and it's very specific. This is not a haphazard process. It's amazing to me that it can happen with such specificity in our cells, millions of times every second in our bodies. You'll see here that in metaphase, what's happening is the microtubule spindle fibers are orienting the chromosomes in, they call it a plate or a line across, and you can see it right here. All the chromosomes and their sister chromatids are in a line across, and they're going to be pulled in anaphase, the middle picture here, to either side of the cell, and then in telophase, it's kind of the last process of mitosis, and then cytokinesis is actually the division of the cytoplasm. Well, here's metaphase in a little bit more detail. Here again is the metaphase plate. We see some other players. The centrosome, I see some microtubules, I see sister chromatids. Over here on the left-hand side, this is just a micrograph or a microscopic picture of mitosis taking place, specifically metaphase. Please try in your mind, press pause and try to circle the asters, draw arrows indicating the movement of chromosomes once anaphase begins. Well, this may be something that you thought of in your mind. Let's talk a little bit more detail about anaphase. In anaphase, it's my favorite phase of mitosis. I don't know why, but it's when the sister chromatids are pulled to either side of the cell. It's a pretty active, exciting phase. Maybe that's why I like looking at it under the microscope. Understand that the microtubules shorten by depolymerization, or just the breaking off of these tubulin subunits at the actual kinetic core. So you've got these centrosomes at either end of the cell. They're not pulling, if you will, the sister chromatids to either side. What's happening is on the rope is shortening, and as the rope it gets shortened and it gets pulled back, and you can see that happening right here in the middle. The chromosome that's attached to it is actually going with the shorter rope towards either side of the dividing. Make sure that you answer this question. Which of the following best describes the kinetochore? Please press pause. Here's cytokinesis. Cytokinesis, again, is the division of the cytoplasm. It's the last step of the mitotic phase. And if you notice here, in cells without a cell wall, like our eukaryotic cells, what happens is something called a cleavage furrow occurs. And cleavage occurs, and it just means the pinching. There's a pinching inward of the outer phospholipid bilayer, and it's just going to divide into two new daughter cells, and you can see that here on the right-hand side. But in plant cells, it's different. Plant cells do not have a malleable phospholipid like fatty bilayer. Well, they do. They have a cell membrane, but they have a cell wall on the outside, so they can't be doing all this pinching and stretching like an animal eukaryotic cell can. And so what you see here is that a cell plate will form in between the two new daughter cells. So let's watch a movie on mitosis.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action that stretches the cell. The chromosomes then unwind and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Cleavage furrow. Make sure you try to answer this question on page 245, please. Thank you in Swahili. And here, try to identify as many of the different phases of mitosis as you can on this picture. Metaphase, prophase, anaphase. Here's some examples. Please answer these objectives. Thank you for your attention.